Have you ever wondered how your body fights off infections? It's a remarkable system working tirelessly behind the scenes. We call it the immune system. It's our internal defense force, constantly patrolling and protecting us from a barrage of microscopic invaders. Imagine a microscopic world teeming with activity. This is the world of our immune system. White blood cells, the soldiers of this microscopic army, are constantly on the move. They patrol our bloodstreams and tissues, always alert for signs of danger. These cellular sentinels are the key players in a complex molecular drama that unfolds every time a foreign entity enters our body. The immune system's weapons are not swords and shields, but a vast arsenal of molecules, each tailored to target specific threats. These molecules, including antibodies and signaling proteins, are the products of a complex interplay of genes, proteins, and cellular processes. Before the immune system can mount an attack, it needs to know who the enemy is. Enter antigens. Antigens are like molecular wanted posters that the immune system uses to identify foreign invaders. These invaders could be anything from viruses and bacteria to fungi and parasites. Each of these pathogens carries unique antigens on their surface, acting like molecular fingerprints that distinguish them from our own cells. Exogenous antigens originate from outside our bodies, like the proteins found on the surface of a flu virus. Endogenous antigens, on the other hand, are generated within our own cells. This can happen when our cells are infected by a virus or become cancerous, displaying abnormal proteins on their surface. The immune system focuses on specific regions called epitopes. These small regions on the antigen surface are what antibodies, the immune system's specialized weapons, latch onto. Antibodies are the immune system's precision-guided missiles, exquisitely designed to seek and destroy foreign invaders. These Y-shaped proteins are produced by specialized white blood cells called B cells. Each antibody is uniquely crafted to recognize and bind to a specific epitope on an antigen, forming a highly specific antigen-antibody complex. The two arms of the Y contain the antigen binding sites, which are like molecular locks that perfectly fit the key of the epitope. This binding event is the first step in neutralizing the threat. The stem of the Y determines the antibody's class and function, dictating how it will interact with other components of the immune system. Antibodies employ a variety of mechanisms to neutralize pathogens. Neutralization involves blocking the pathogen's ability to infect cells. Opsonization involves coating the pathogen with antibodies. This tagging process makes the pathogen more visible and appealing to phagocytes, which engulf and destroy the marked invaders. The immune response is a beautifully orchestrated cascade of events, a carefully choreographed dance of cells and molecules designed to eliminate threats and maintain the body's equilibrium. The first act begins with recognition. Immune cells constantly patrol the body, on the lookout for anything suspicious. When a foreign antigen enters the scene, specialized cells called antigen-presenting cells engulf the invader, break it down, and present fragments of the antigen on their surface. T cells, the immune system's generals, inspect the presented antigen fragments. If they recognize the antigen as a threat, the alarm bells start ringing. The second act is activation. T cells, now activated, differentiate into specialized subtypes, each with a specific role to play. Helper T cells act as coordinators, releasing signaling molecules called cytokines that rally other immune cells to the fight. Cytotoxic T cells, the assassins of the immune system, are tasked with directly eliminating infected cells. To understand the immune response, we peer into the microscopic world of cells and molecules. Molecular techniques are key. ELISA detects specific antigens or antibodies. Western blotting identifies specific proteins. Flow cytometry sorts cells by size and protein expression. One of the most remarkable features of the immune system is its ability to remember past encounters with pathogens. This immunological memory is the cornerstone of acquired immunity, allowing our bodies to mount faster and more effective responses upon subsequent encounters with the same threat. This remarkable feat is accomplished by specialized cells called memory cells. After the initial encounter with an antigen, a subset of activated B and T cells differentiate into memory cells. These long-lived sentinels remain in circulation or reside in lymphoid tissues, ready to spring into action should the same antigen reappear. Upon re-exposure to the same antigen, memory cells can rapidly proliferate and differentiate into effector cells. This rapid response ensures that the pathogen is eliminated before it can establish a foothold and cause disease. The speed and efficiency of this secondary immune response are the reason why we rarely get sick from the same pathogen twice. Vaccines are one of the most transformative inventions in human history, saving millions of lives and eradicating deadly diseases. They are the epitome of preventive medicine, harnessing the power of the immune system to protect us from future infections. Vaccines work by mimicking natural infections, tricking the immune system into generating a protective immune response without causing the actual disease. Vaccines typically contain weakened or inactivated forms of the pathogen or even just specific antigens. These harmless versions of the pathogen are enough to stimulate the immune system's recognition and activation phases, leading to the production of antibodies and memory cells. When a vaccinated individual encounters the actual pathogen, their immune system is primed and ready to respond swiftly and effectively. The memory cells generated through vaccination are on high alert, ready to mount a rapid and robust secondary immune response, preventing the pathogen from causing disease. While the immune system is a powerful defender, it's not without its flaws. Sometimes the immune system can misidentify its own cells and tissues as foreign, launching an attack on the very body it's supposed to protect. This immune system malfunction is the root cause of autoimmune diseases, a diverse group of chronic conditions that affect millions worldwide. In autoimmune diseases, the immune system's recognition system goes haywire. Immune cells, mistaking self-antigens for foreign invaders, launch an assault on the body's own tissues and organs. This misguided attack can manifest in a wide range of symptoms, depending on the target organ or tissue. For instance, in type 1 diabetes, the immune system attacks insulin-producing cells in the pancreas, leading to high blood sugar levels. In rheumatoid arthritis, the immune system targets the lining of the joints, causing pain, stiffness, and inflammation. The relationship between the immune system and cancer is a complex and multifaceted one, marked by both conflict and cooperation. While the immune system plays a crucial role in recognizing and eliminating cancerous cells, cancer cells are notorious for their ability to evade and even manipulate the immune system to their advantage. The immune system is constantly on the lookout for abnormal cells, including those that have undergone malignant transformation. Immune cells, particularly cytotoxic T cells and natural killer cells, can recognize cancer cells as foreign due to the presence of tumor-associated antigens on their surface. These antigens, often resulting from mutations or abnormal gene expression, act like red flags, signaling the immune system to eliminate the rogue cells. However, cancer cells are masters of disguise and deception, constantly evolving and developing strategies to evade immune detection and destruction. They can downregulate the expression of tumor-associated antigens, making them less visible to the immune system. They can also release immunosuppressive molecules, creating a microenvironment that suppresses immune cell activity. The field of immunology is rapidly evolving, fueled by groundbreaking discoveries. These discoveries are reshaping our understanding of the immune system. From personalized immunotherapies to innovative vaccine platforms, the future of medicine is intertwined with our immune system. Cancer immunotherapy harnesses the immune system to fight cancer. Immune checkpoint inhibitors have shown remarkable success. CAR T-cell therapy involves modifying T cells to target cancer cells. Immunotherapies hold promise for treating autoimmune and infectious diseases. Researchers are exploring mRNA and DNA vaccines for speed, efficacy, and safety. The immune system is a testament to the elegance and complexity of biology, a symphony of molecular interactions that orchestrate our body's defense against a constantly evolving world of pathogens. Understanding the molecular basis of immunity is not just an academic exercise, it's the key to unlocking new therapeutic avenues and improving human health. As we delve deeper into the molecular world, we begin to appreciate the intricate interplay of genes, proteins, and cells that govern immune responses. We unravel the secrets of antigen recognition, antibody diversity, and immune memory, gaining insights into the delicate balance between immune activation and suppression. The immune system is not just a collection of cells and molecules, it's a dynamic and interconnected network, a microcosm of the body's intricate workings. By understanding the molecular symphony of immunity, we can begin to compose new melodies of healing, orchestrating the immune system's power to protect us from the microscopic world that surrounds us.